We have had a September holidays break, right? So I will try to revisit a bit of what I covered previously, and on the second part of the lecture notes on DA, which is titled DNA Analysis and Genomics, one of the very big concepts that we covered or that I taught you on was called RFLP. So can you guys tell me, to the count of 3, what is RFLP? 1, 2, 3. Okay, I give you a clue. Restriction. Okay, again, wait. 1, 2, 3. Restriction. Fragment. Length polymorphism. Okay, very good. Restriction, fragment, length polymorphism. Okay, the other thing is, this is at page 19 of your lecture notes. Can everyone write down? In exam questions, many students mistakenly write about RFLP as if it is a technique. Okay, so I want you to write at this concept map at page 19. RFLP is not a technique. Okay, RFLP is not a technique. And then you write down, it is a concept that is used to distinguish different sequences of nucleotides. RFLP is not a technique. It is a concept that is used to distinguish different sequences of nucleotides. Okay, everyone okay? Now, the different sequences of nucleotide as shown over here can arise due to number 1, a mutation. So over here, single nucleotide polymorphism. So there might be a mutation, but the mutation must occur at a restriction site. Very good. Here, must occur at a restriction site. If it doesn't occur at a restriction site, then we say it's not an very good, not an RFLP. The other one, so number 1, it can be detected via mutation, differences in nucleotide sequence. Number 2 is RFLP can also be detected via the different number of repeat sequences that you and I have. Each of us have different number of two repeat sequences. 2 types of repeat sequences. Can all of you remember what are they? One of them starts with S, one of them starts with V. The S is very good, STR. What is STR? Short tandem repeats. Very good. The V is VNTR. What is VNTR? Variable number of tandem repeats. Which one is longer? VNTR. Which one is shorter? STR. Okay, very good. And again, the different number of repeat sequences must be found between, very good, must be found between restriction sites. If it's not found between restriction sites, then we say it is not an, very good. Everyone clear? Okay, good. By the way, class 13, where's Junya? Okay, it's okay. Can Louisa help tell Junya I want to see him at the end of lecture. It's about the class leader thing. Something about class leader. Okay? Okay. So I continue. Now the procedure when you do RFLP, there are the procedures to carry it out. The PCR, is it a must to do PCR at the beginning? It's not. If you choose not to do PCR, then what becomes a must later on? Southern blotting. If you have done PCR initially, then southern blotting becomes very good. Okay, then after that I asked you guys to do this activity. This is page 19. Sorry, page 20, 22, 25, 22. Okay, this is at page 22. Okay, so can I give you a quick 5 seconds? Before I give you a quick 5 seconds, can all of you remember what was the clue I gave all of you? What did I tell you to take note when you do this question? Where the probe is. Very good. So all of you, I want you to write down again. You write down exam tip. Take note of where the probe is. Then can I give you a quick 5 seconds? You buzz with your neighbor what your answers of where the bands will be are. Quick 5 seconds from now. Okay, everyone, can I have silence and look at the front? Okay, now what happens is that here it says the first thing to take note, there are two different types of alleles, allele 1 and allele 2. What are alleles? Very good. I heard from some girl here. So I want you, a good girl here. I want all of you to write down alleles are different forms of the same gene. Different forms of the same gene. Then you can write there example big A and small a alleles, right? So it's an A gene, but there's big A, there is small a as well. And I've gone through with you this definition previously. Okay, so allele 1 and allele 2 over here in this diagram, it shows that they are different in terms of the number of restriction sites. Very good. So also I want you to write down allele 1 and 2, you write down allele 1 and 2 are different in terms of number of restriction sites. Okay, so I'm not going to write down because my page always runs. Okay, allele 1 and 2 are different in terms of number of BAMH1 restriction sites. So allele 1 has how many BAMH1 restriction sites? Edward, right? What? Edward. Benjamin, you cut your hair. Is to study for your promos, is it? Or that's a sacrifice. Okay. Okay, so either Benjamin or Edward, how many BAMH1 restriction sites are there in allele 1? 3, very good. So there is 1, there is 2 and 3 over here. Allele 2, how many restriction sites are there? 2. So when there are 2 restriction sites, okay, everyone, when there are 3 restriction sites, how many fragments will be generated? When there is 2 restriction sites, how many fragments? 1. Okay, we will take note, the 1 fragment is going to be a long one of 9 kb. It's quite heavy. But here, the 2 fragments is 5 kb and 4 kb. Okay? So, okay, now here, can all of you write down, what is this column called? DNA? Start with very good. Very good. Ladder. DNA ladder. What is DNA ladder? Equal. I don't have the official textbook. You can always refer to your lecture notes. But I want you to write DNA ladder consists of fragments of DNA of known sizes. Known size. So here it is, you already told at this band is 10 kb. This band is 5 kb. This band is 2 kb. Okay? Okay? Joanna, okay? Yes, okay, good. Okay, so for allele 1, how will you draw the band? Allele 1 has 2 fragments, right? 5 kb and 4 kb. Right? So there will be allele 1 will have one band here at 5 kb and maybe another one here at 4 kb. Follow? But allele 2 has one fragment at 9 kb. So it should be, it will run a bit beyond the 10 kb band. Everyone follow? Any questions so far? Okay, these are all the bands that will be present upon gel electrophoresis. Write that down. Okay, write that down. These are all the bands that will be detected after gel electrophoresis. However, this question asks you, with the addition of the probe, draw the position of the DNA after southern blotting. Southern blotting would have nucleic acid hybridization, am I right? With the probe, right? The probe and the target sequence. Am I right? So with the probe, and that's why I said the exam tip is to take note of where the probe is. So if you take a look, this is where the probe is. So for allele 1, the probe binds to which fragment? 4 kb. Does the probe bind to 5 kb? No, which means that after southern blotting, do you think the 5 kb band will appear? No. So after southern blotting, you will not see the 5 kb band. You will see only the 4 kb band, which means that is going to be like this. It's going to be like this. Only the 4 kb band. Okay? But at the allele 2, okay. Allele 2, the probe is over here and therefore, will it detect the 9 kb? The probe is over here. Will it detect the 9 kb? Yes. So for allele 2, it will still be 9 kb. Everyone follow? Okay. So class, this is very important. Something you learn. The bands look differently after gel electrophoresis and after nucleic acid hybridization. Okay, can you say to your neighbor? The band pattern looks different after gel electrophoresis and nucleic acid hybridization with probes. Okay, after you say to your friend, you write down, the bands look different after gel electrophoresis and nucleic acid hybridization. Sorry, on top of that, I want you to write there what I learned from this activity. What I learned from this activity is that the bands look different after gel electrophoresis and nucleic acid hybridization when I add probes. Number two, you write down, usually the bands after nucleic acid hybridization with probes is lesser than the number of bands present after gel electrophoresis. Number 2, usually the number of bands after nucleic acid hybridization with probes are lesser than after gel electrophoresis. Okay? Write down the 2 points. If you didn't get it, you ask from your friend. Okay? Can everyone clear? Mao, are you clear? Okay, good. She said her name is Mao, serious. Okay. So then class, just to remind you again, RFLP has 3 applications. So the 3 applications are here: DNA fingerprint, disease detection, and genome mapping. Mrs. Lim can see at the back? Can, okay. Yeah, Mrs. Lim is back. Let's give a round of applause for Mrs. Lim. Okay, we'll take good care of her. Okay, DNA fingerprinting has 3 further applications, right? I want you to write down. Number 1 is, can someone remind me? Number 1 is it parentage? Huh? Oh, criminal, yes, criminal. Criminal cases. Number 2 is parentage. Number 3 is homology, right? Number 3 is homology or closely related species. Okay, everyone clear? So DNA fingerprint has 3 further applications, right? Okay, now over here, even before I go on with the lecture, I ask all of you to do your tutorial question, am I right? Okay, can all of you take out your tutorial? John, did you do your tutorial? No, why never do? Forgot. Did you get a message from your bio, right? Yes? Yeah, this morning, sorry, because I sent her at 11:30 at night. Okay, so she is excused because it was my mistake. Okay. Okay, so everyone, can I have silence? Now, in your tutorial question, I still don't have silence. Okay, at B part 2, the previous question was on PCR. So here it says how PCR facilitates genetic fingerprinting, right? Now, the question is how PCR facilitates. Facilitates means, I want you to write how PCR makes genetic fingerprinting easier to be done. Because the bands can be more easily visualized. I'm asking you to write how to interpret the question, right? So I want you to write down how PCR facilitates genetic fingerprinting. This question, right, is how PCR makes genetic fingerprinting easier. Drashi, write down. How PCR makes genetic fingerprinting easier by making the bands by making the bands easier to be visualized. How does PCR help to make the bands easily visualized? You and Tian, I'm asking you to write. This is tutorial. B part 2. DNA analysis and genomics. 1B part 2. Okay? So the answer, right, H2 class, I want you to write, actually I find the answer missing one word, so I want you to write PCR is very sensitive because it can detect very small amount of original DNA. Okay? No worries, I will send you the answer as well, so you take key notes, key ideas of the answers first, okay? I'm not giving you time to copy down. You can't see, is it? Then where's your glasses? Then take it out lah, put it on. Again, you remind me of my son. I'm always yelling at him, where's your glasses? Okay. Then the next thing I say to my son, is your glasses clean? Clean, okay, go. Okay. Okay, so class, I want to see the word sensitive. Sensitive because it's very small amount. It can detect very small amount. And class, once it detects a very small amount, what does PCR do to that DNA? It can detect just one sequence. What will it do? Amplify. So the subsequent keyword is amplify. Make many copies, and that's how it makes analysis easier because the bands will be very visible. Okay? So 2 marks, any 2 of these points will be valid. Okay? Okay. So I'm not giving you time to copy down, okay? Later I'll send you the answers as well. So I move on. So this was a question on genetic fingerprinting, but it relates to PCR. Right? How does PCR help it to be better? Okay, now let's take a look at C. C, the examiner's comment says, we have to read the preamble given very carefully first. Okay, so let's do that. The law in many countries don't allow the import and export of endangered species. What is endangered species? I don't have an official definition, but it refers to species of animals which are almost extinct, in the threat, has the threat of being extinct in the future, which means their numbers are already declining very rapidly. And almost all species of animals are actually under threat because the Amazon forest is shrinking. Okay, but anyway, that's a different lecture altogether. Okay. No, we don't have a lecture, but I'm also passionate about endangered species. Okay, so anyway, here the question says, how the process of genetic fingerprint, explain the process of genetic fingerprint. So everyone, I want you to write down, there are 2 parts to this question. Number 1, you have to explain genetic fingerprint. Right? There are 2 parts and total 5 marks. So let's take a look. How do you explain genetic fingerprint? We isolate the DNA sample from the organism first. Right? And genetic fingerprint uses what concept? RFLP, very good. Uses the concept of RFLP. So we say RFLP analysis, right? The word is RFLP analysis. Is done by, then you tell me the steps. Number 1, cut with restriction enzyme. Number 2, run your gel electrophoresis. Number 3, do Southern blotting. Okay? What Hatiang? What did she say? No? Okay. Okay, so the last one is Southern blotting. And H2 class, I want you to see how Southern blotting is described very succinctly. There are essay questions that ask you to describe Southern blotting only, and then they give you 5 marks, so you have to write a lot. Sometimes Southern blotting is only part of the answer, so it has to be very succinct. So you see the keywords is, remember I told you when you talk about probes, there are a few things to mention. You must mention how it's labeled. You must mention it is single-stranded. You must mention the probe is DNA. 3 things to mention. And mention that the probe is complementary to the target sequence, to the RFLP loci, and is detected by autoradiography. So the method of detection will be complementary to how your probe is labeled. Everyone okay? Okay, over here, you okay? Okay. Okay, some students ask me, Madam Reshma, if I wrote about PCR, I do PCR first, I say also can. So you can write PCR was done first, but then your Southern blotting would then be, you write that Southern blotting is optional and it can be carried out via blah blah blah, SS, radioactive DNA, etc. Okay? Okay, so I have addressed the first part of the question, which is explain genetic fingerprinting. Everyone okay? Now is the second part, which is the slightly tougher part. And the examiner's comment says that many students misinterpret this second part. So you have to listen to me very closely. Okay. The second part says, suggest how genetic fingerprint can be used to verify whether the endangered species organism. Okay, let's say for example it's a tiger, right? The tiger is endangered species. Whether the tiger is bred in captivity. Okay, so everyone, I want you to highlight bred in captivity. Then I want you to put 3 stars here. Okay. Then I want you to write down what the examiners say, that what the examiners comment on how the students misinterpret. Okay. So I want you to write down genetic fingerprint here is used to test whether the animal was bred in captivity or in the wild. Write down. Genetic fingerprint is used to test whether the animal is bred in captivity or in the wild, comma, not whether it is endangered or not. Not whether it is endangered or not. Okay? So endangered it definitely is. There's no issue about that. But it's just whether the species was born and bred in the wild or in captivity. Everyone okay? Okay, before I show you the answer, can you give me a one-liner answer? How do we use genetic fingerprint to find if the tiger, for example, tiger was bred in captivity? How do we do that? Remember when we do genetic fingerprint, what must we do? We will get a few DNA fingerprint profile, right? Unique profile. Then we will compare. So the issue here, if you are trying to find if the tiger is bred in captivity, you have to compare with another tiger that was also correct. That was also bred in captivity. Okay, so that's the answer. So the first point is that wild and captive have different genetic profiles, have very unique DNA fingerprint. Then number 2, we compare the fingerprints with those of the captive individuals. Okay, with those here, this one is captive individuals. And if the endangered species is bred in captivity, there will be a match in the bending pattern. We compare the bands, and if there is a match, then the tiger was bred in captivity. If there is very little match, then most probably the tiger was bred in the wild. Everyone okay? Okay, any questions? Jalil? No. Yeah. Sorry, I can't hear. How do they have a difference in the genetic profile? Okay, there's a student here who asks how do the wild and captive animals have different genetic profiles? Okay, the reason is because in the wild, the species will have a higher level of genetic variation because they have more possible mates to breed with. But in a captive, it is lesser genetic variation because it is usually a high level of what we call inbreeding. Okay? Okay? Okay, so I move on to the next question. I hope all of you have taken note. The next question is on gel electrophoresis and RFLP, and the application of RFLP is disease detection. Okay, disease detection. Everyone ready? Okay, here it says the DNA of a family affected by rare autosomal. So we are told it's autosomal. We are told it's autosomes. Okay, it's autosomal, and you also told it's rare. Now, when you are told that the disease is rare, it might be, it can be a clue to tell you that the gene is recessive or dominant. Recessive. Okay, here is it's not confirmed. It's not confirmed. We have to confirm it later in the question. But it seems as if it is cluing us to that it's recessive. Okay. Then the other thing for you to take note is that it says the two genes A and B are found on chromosomes 12 and 23. What is this trying to tell you? 12 and 23. Okay, if I may go back to your lecture notes, under disease detection application of RFLP, remember I ended the last lecture by explaining to you that to detect a disease causing allele, there are two instances where the RFLP can be used to determine. Okay, and these are the two cases. So number one is RFLP is found within the disease causing allele. Number two is if the RFLP is very tightly linked to the disease causing allele. Right? Okay. So over here, do you remember what I told you was the definition of linked genes? Two genes are linked if they are located on the same chromosome. Very good. Now let's take a look at the tutorial question. In the tutorial question, is it linked genes A and B? No. So right here, so you write A and B are not linked genes because they are located on different chromosomes. Okay. Okay. Okay, then the other thing is they also told you the restriction sites. So A1 and A2 are different alleles of gene A. B1, B2, B3 are different alleles of B gene. Right? So you guys take note. Here is the restriction. Here is the RFLP, the one with the asterisk. Polymorphic restriction sites are indicated. Okay. Okay, so A1 is 10 kilobases and it's one fragment. So write that down. A1 is 10 kilobases, one fragment. What about A2? A2 is how many bases? How many fragments? 2 and what are the sizes? 3 and 7 kilobases. You write there 2 fragments. Can I give you a quick 5 seconds to do that for B as well? B1, B2 and B3. Okay. So B1, how many fragments? 1. What's the size? 5 kb. B2, 2 fragments. What is the size? 2 and 3. B3, how many fragments? 2. What are the sizes? 1 and 4. Okay, very good. So as I go on down, all of you must remember the fragments and the sizes, okay, of the different alleles. If you can't remember, you refer to this diagram in your tutorial. The other thing for you and I to take note is this, the probes. But the thing about the probes is that the probe is everywhere in the entire A allele and in the entire B allele. Okay? Do you all realize that? So I want you to write, since the probe, okay, I'm not going to write. I say you write. Since the probe hybridizes to the entire allele sequence, comma, all fragments of the allele will be visualized. Write down. If the probe hybridizes to the entire allele sequence, all the allele fragments, all the fragments will be visualized. You guys follow? Okay, good. Now we are ready. Okay, with reference to figure 4.1, how two alleles of gene A can be distinguished using jet electrophoresis. So, can anyone tell me how do we distinguish the two alleles of A1 and A2 when you do jet electrophoresis? They will give different bands. Very good. So A1 will give how many bands? How many bands? 1. A2 will give 2 bands, and you tell me the size. Right. So that's the answer. A1 will give one large fragment of 10 kb, and this is the part where you cite data. You must cite data because it says with reference to figure 4.1, and A2 has 2 fragments of 3 and 7 kb. Okay, so I think this part is quite simple. Okay. Okay, I move on to the subsequent part. This part is where I want all of you to write. Now this diagram given to you here, is you write there a genetic fingerprint. A genetic fingerprint, and it is unique for every different individuals. Okay. Okay, so here it says the pedigree chart of a family affected by the disease, right? So H2 class, I want all of you to highlight this word. It says phenotypically normal parents. Am I right? Phenotypically normal couple. What is phenotype? You may not be very clear, so I want you to write down. Phenotypes means, I cannot remember the exact definition, but I think it's sufficient for you to write down all the expressed traits. All the expressed traits. That means whether you have the disease, whether you don't have the disease, what hair texture you have, what eye color you have, etc. Okay? So phenotype is all the expressed traits. So over here, actually, when they say phenotypically normal, it's a clue, right? Because if you take a look at the parents are normal, right? Because only if it's shaded, then will the person have the disease. The parents is phenotypically normal, but they in order to have a child W that has the disease, the parents most probably have the recessive gene. Carrier, very good. I heard a girl say carrier. So I want you to write down here, parents are carriers. Parents are carriers of the recessive disease allele. Recessive disease allele. Okay? Okay. Now, the next thing what I want you to do is, can I give you a quick 10 seconds? I want you to write the genotype. That means which alleles do this person have? The father, the mother, W, X, Y, and Z. Okay, I'm going to do for the father to illustrate to you how it's done, then you will do for the rest. Okay? Okay, for the man, if you take a look, he has 10 kb, he has 7 and 3 kb. 10 kb is A1 or A2? A1. So he has A1 and 7 and 3 is A2. So his genotype is A1, A2. Remember, we have 2 alleles for every gene because we inherit one from mother, one from father. Okay, so the father is A1, A2. What about the B? He has 5 kilo bases. 5 kb would be B1, B2 or B3? B1, and 2 and 3 would be B2. So this is his genotype. So his genotype is A1, A2, B1, B2. Okay, can I give you quick 10 seconds to do for 2, 3, 4, 5? Sorry, 2, 3, 4, 5. Okay? Okay, can you guys tell me together as a cohort? Okay, number 2, what is the A allele? A2, A2, very good. What about the B? B2, B3, okay, very good. For number 3, the A allele? A1, A2, what about the B? B2, B2, okay, good. Number 4? A2, A2. Then for the B? B1, B3, am I right? Correct, okay. Number 5, A? A1, A2, B? Okay, 6 is we don't know yet. Later they will ask us what we think. Can everyone okay? Anyone lost? Okay, good. Okay, so class, from these band patterns, the next question I'm going to ask you is, we know lane number 3 is the poor thing. Lane number 3 has the disease, right? Now the question is it the A gene or the B gene which is the disease gene? Which one? B, okay, very good. Wow, synonymously B. Why B? Okay, B2. B2 is specifically the recessive problem allele, right? Okay, because B2 is quite easy because this person has 2 copies of B2. And so I want all of you to write down recessive copies need to be present in 2 copies in order for the trait to be expressed. Write down. Recessive alleles need to be present in 2 copies in order for the trait to be expressed. Okay?
What's your name? What's your name? Sorry? Chloe. Chloe, you don't have your tutorial, is it? Okay, you can take a rough piece of paper to copy because you are also very sleepy, I think. You need to wash your face? Wake up already, okay, good. The attention.
Okay, so as I said, very good. You guys are able to tell me B2 is a recessive allele. But why not A? Why isn't A1, A2 a disease allele? Because the father also has the same genotype A1, A2 and the father is fine, is phenotypically normal, is healthy. Okay, very good. And in fact, you guys have just told me the answer to, what is this? A part 1. Which gene causes the disease and the reason. Right? And the reason. Okay, sorry. Part 2 says the genotype, state the genotype of child W and explain the inheritance of the disease. Okay, so child W is lane number 6 and we are supposed to state the genotype in terms of A1, A2, B1, B2, B3. Okay, over here, you are told that Z also suffers from, the genotype of child W. So W is here. So it's A1, A2, B2, B2. Right? So here. And explain the inheritance is that W is homozygous for B2 allele, and because one allele is inherited from one parent and disease is recessive. So here is where we confirm that the disease is recessive, right? When we saw rare, in our mind we say, it could be recessive. When we saw the fingerprint, it confirms our suspicion. Can everyone follow? Okay, good. Okay, now there's a fourth child, Z. Now, Z is homozygous at both gene loci. So I want you to highlight homozygous. Right? Now, for those of you who may not be as familiar, what does homozygous mean? Homozygous means, I can't remember the official definition, but I think you can write down same two alleles. Same two alleles. Write down. Homozygous means same two alleles. Heterozygous means different alleles. Okay, very good. So it's the same allele, and they're asking what is the bands for child Z. Okay. So how do we do this? Is that I want you to write down at this question the concept that you need to remember. Okay, you write down at part C. At part C, right? I want you to write down every child inherit one allele from mother and another allele from father. You follow? Angeline, is it? Part C, you write down the concept is every child inherit one allele from mother and one allele from father. Susan? Okay, so over here, after knowing that concept, we also must remember that for this child Z, the allele that he or she got from mother and father must be the same. Must be the same. Right? So when you bear in mind these two concepts, then the only answer is the A allele has to be what? A2. Right? Only A2 is present in one copy from father, one copy from mother. And for the B allele, it has to be B2 as well. Everyone follow? Ken? Cynthia, you okay? Okay. So the answer is the gene A should show 7 kb and 3 kb. Gene B should show 3 kb and 2 kb because the genotype is A2, B2. Okay? Any questions? Okay, part D, 2 features of a gene probe. I think this one should be quite easy for you. Remember, I told you whenever you mention probe, you must mention number 1 that it is SS. Number 2, the probe is DNA or RNA. Number 3 is how is it labeled. Then maybe you can add number 4 is that it must be complementary to the target single-stranded molecule. Okay? Who's coughing very bad? Kylie? No. Okay. I hope you get well soon. Okay, part E, RFLP is found on the same chromosome as the disease-causing allele. Okay, so everyone here, I want you to highlight found on the same chromosome. So if it's same chromosome, it means the RFLP and the disease-causing allele are linked. Then you put 2 star. They are linked. Explain how the RFLP can be used as a genetic marker to detect the disease. Right? So when they are linked, how RFLP can be used as a genetic marker to detect the disease. Okay? Now over here, I'm going to go back to the lecture notes. and quickly recap on the disease. Actually, I've already covered this in the previous lesson, but I will quickly go through with you. Okay, so class, in order to detect disease-causing allele via RFLP, there are two possible ways. Number one, if the RFLP is found within the disease-causing allele. Okay, that one I will go through a bit later. Number two is, which the tutorial question is asking, is when the RFLP is tightly linked, right, to the disease-causing allele. So let me teach you again on RFLP being tightly linked, and it's over here. Okay. Now, at this page, I also taught you the concept of when we say two alleles are linked. I told you the difference between A and B. Can you guys tell me? When they are linked, they can be two different types of links. Very good. Completely linked, which is A. A is the A and the E genes. The A and the E genes are completely linked. And here in B, the A and E are incompletely linked. Incompletely linked. Okay? So, now, if they are completely linked, that means what cannot occur? Crossing over cannot occur. Then what else did I say? Therefore, A and E are always inherited as one unit. Inherited as one unit. Okay? And if they are incompletely linked, that means what can occur? Crossing over can occur. But I also told you that in order to detect the disease, which of these must the RFLP be? Completely linked or incompletely linked? Completely linked. So here, in order to detect disease, it must be, the RFLP must be completely linked. I'm not going to spell out, okay? The RFLP must be completely linked in such a way that the RFLP and the disease allele will be inherited together as one unit. Okay? So over here it shows you that the disease-causing allele and the RFLP marker is always inherited together as one unit because they are very close to each other. Okay, so therefore, if the RFLP marker is detected, that means the individual has the disease-causing allele. Everyone follow? And another thing for you to take note is this case of the RFLP being very tightly linked is very useful when the disease allele is unknown. We don't know what is the disease allele, but we know it is very tightly linked to this RFLP. You guys follow? Okay. Okay. Sorry, just give me a few more minutes. A and E, when the genes are incompletely linked, I want you to write down that this concept I will explain to you in another lecture is useful in genomic mapping, which is the last application of RFLP. Okay? If it's incomplete link, it will be very useful in genomic mapping. So if you take a look at the answer says that RFLP will serve as a marker if it is tightly linked to the disease-causing gene, and they are always inherited together, and if the RFLP is detected, that means the individual has the disease allele as well. Everyone follow? Okay? Okay, class, hang on. Some last final announcements.